I'm Brent Delmonte, the Vice President for Federal Government Relations at BIO, which is the Biotechnology Industry Organization, representing roughly 1,100 members, hundreds of companies, most of which are smaller in size, working on their first product for delivery. But we have our company size runs the gamut from large to small, and very happy that you joined us today for a little lunch briefing we're having entitled The Role of Biotechnology in Biodefense Preparedness, where you can learn more and more about what biotechnology and its companies are doing to search for different cures and treatments for things which we hope will frankly never have to be used, for the fact that they would be countermeasures for a number of different bad events. We're going to hear from experts today on the need to prepare and protect the nation's health and security, and the value that the biotech industry can provide in that critical preparedness. Biotechnology industry has been and continues to be developing innovative products that can simultaneously advance public health and enhance our nation's preparedness and security. Much has been accomplished, but much more needs to be done. First, you're going to hear from Rich Hamburg, who's Trust for America's Health, on the need for enhanced biopreparedness, and then you will hear from a number of recognized experts in the biotechnology field describe the types of innovations and the promise that those innovations hold for preparedness and public health. You're going to hear from these experts the critical next steps that are needed to bring our nation to the level of preparedness that we need, including strong leadership and a long-term commitment, shared risk, and a strong public-private partnership, which really is the name of the game. It's both our industry working with the government to ensure that we will be ready if and when we need the infrastructure to deal with a attack of some sort. We are going to talk a little bit about the need to better prepare our country in terms of helping our companies get to what we call the valley of death. This is the portion which we call this cycle right there. It's after your early stage funding has run out, but before the government does its purchase under the Bioshield program. You're left in the valley there where you're running enormous, you're going through a lot of capital and you don't have anyone to help you pay for it. That's why we need a better public-private partnership. Last, before I turn it over to Rich, I'd like to thank the sponsors of this event. First of all, I'd like to say thank you to Congressman Rush and Congressman Shimkus, who serve as our house chairs of the Biotechnology Caucus. They are fantastic to work with, and we do appreciate all their help in putting this event together. Also, I'd like to say a special thanks to the sponsors for this lunch briefing today, specifically the Canvas, Alnylam, AlphaVax, Applied Biosystems, Avant Immunotherapeutics, Baxter Healthcare, BioCryst, Elucis Therapeutics, Genvec, Invitrogen, Pharmathene, VaxGen, and Vaccinate. And again, I hope you view this as very worthy. We have great panelists to discuss this matter with you today. I'll turn it over now to Rich Hamburg.
Thanks, Brent. I'm glad to be here today. For those of you not familiar with Trust for America's Health, we're a nonprofit, nonpartisan organization dedicated to saving lives, working to making disease prevention a national priority. After years of underinvestment, our public health system lacks the resources it needs to tackle a full range of health threats, from preparing for potential chemical or biological attacks to addressing the serious chronic diseases like cancer, heart disease, responding to emerging infectious diseases such as avian influenza. Trust for America's Health recommends that the United States prepare for bioterrorism on a systemic level, with policies able to address any major health outbreak that appears, be it biological, chemical, radiological, or natural disease outbreak. This all-hazards approach must replace the current threat-of-the-moment mentality that drives federal programs and funding in large part. Failure to do so is what results in public health catastrophes such as the inability to provide basic health to the victims of Hurricane Katrina. This past December, we released the latest edition, I believe the third, of our Ready or Not 2005 report on the nation's public health emergency response capabilities. Overall, our report found a lack of accountability on which to measure federal bioterrorism preparedness. While an expert panel convened by TIFA acknowledged that significant progress has been made post 9/11, overall experts gave federal public health and bioterrorism preparedness performance a grade of D+. It's D, not B. Let me summarize some of our key recommendations. In the area of accountability, federal government in consultation with states and others should develop annual performance measures for preparedness. This includes the federal government establishing a limited number of measurable and achievable priorities that reflect basic preparedness, including measurements of things like strategic national stockpile status, improving lab capacity, and those sorts of things. These priorities should be set through a public consultative process with the states, localities, and the public health community. Each year, each state should be able to measurably demonstrate that they're at a targeted level of preparedness in these particular efforts. And that'll work for bio-preparedness, or now the funds that are out there for pandemic flu preparedness. In addition, federal government should mandate things like tabletop exercises to test basic preparedness at the state level in various categories, with requirements to report back with after-action reports to the CDC, and then allowing the CDC to analyze that data and make recommendations, best practices, those sorts of things. On the topic of leadership, preparedness should derive from the highest levels with accountability from the Department of Health and Human Services secretary level and the governor's level. Performance measures should be provided via a memorandum of understanding between the states, between the governors, and the Secretary of Health and Human Services for all preparedness grants from the agency. In addition, DHHS should have a single senior official accountable for all public health programs, including chronic and infectious diseases and emergency preparedness. This official should have the authority to coordinate programs, determine budgets, and make personnel decisions. Not currently the case. Thirdly, on surge capacity, federal government should ensure that all communities have surge capacity to respond to bioterrorism or other public health emergencies. This includes adequate, effective, and properly equipped public health workforce ready for day-to-day public health concerns, as well as larger scale emergencies. Federal government should more actively recruit and retain volunteer medical personnel under the National Disaster Medical System. Congress should also enact and fund programs to increase the size of the public health workforce capable of responding to bioterrorism and other public health emergencies. We're facing a whole new world of public health threats, ranging from deliberate acts of bioterrorism to new outbreaks of infectious diseases. For some of these threats, known entities like smallpox and anthrax, we have some countermeasures. We should be able to improve the efficacy of these vaccines and medications. Beyond these threats of bioterrorism, the more significant widespread danger break of a new and virulent infectious disease like pandemic flu. Scientists and medical experts do not believe existing vaccines and antiviral medications will be as effective as they should be against new threats. For the moment, there always have, but there's a deep need to improve upon that. We need to strengthen our capacity, develop new and effective countermeasures quickly, and then mass produce those countermeasures when they're needed. These threats may, if we're lucky, never emerge, but we're due for another pandemic. Flu pandemics occur on average 3 times in every century. We must prepare for these awful eventualities as if they were a certainty. This is a challenge for our best engine of innovation in the U.S. biotechnology industry. They have developed treatments and cures for conditions long thought untreatable. But the incentives for this industry to focus on public health emergency response products do not match the need for new countermeasures. These businesses cannot count on research in this area bringing back returns the marketplace and their stockholders demand. They were to commit their resources to countermeasure research, and the threat never manifested itself, they would struggle to stay in business. The federal government needs to provide incentives for business to take on this risk. Such incentives could certainly include tax benefits, guaranteed marketing, even direct federal investment. Any liability protections must go hand in hand with injury compensation as well. The development of these countermeasures is essential for an all-hazards protection. Hopefully we'll never need to use them, and if we don't, their development will likely be beneficial for other areas of public health preparedness. But we must prepare for the worst-case scenario, and that includes increasing research, development, and production of new countermeasures. Additionally, in the area of pandemic flu preparedness, we still have a long way to go in building effective public-private partnerships to assure we're able to respond to a pandemic whenever that might occur. While the latter, much of the federal government's resources are now focused on increasing our capacity to produce vaccines using either egg-based or cell-based technology. While the latter is an important improvement on current technology, we're still operating in a 20th century world, and we should be striving for 21st century solutions. Some of the things that we need to start thinking about: developing a universal flu vaccine, one to be effective against all forms of flu, annual and pandemic. Developing effective rapid diagnostics that can be used in resource-poor settings to determine if a pandemic has begun and whether, to determine if it's been begun. And currently it takes weeks to get a definitive analysis and diagnosis of H5N1 virus. In Indonesia, for example, where we've seen the most disturbing cases. Developed creatively, these same diagnostics could be used during a pandemic as home test kits to determine who should or should not go to work or school and whether or not someone should seek medical care. To achieve these goals, we'll need to require flexibility and creativity of the private sector and the resources of the federal government, or at least enough financial incentives so that we see appropriate levels of investment to assure that public health needs are adequately met, even when the market does not demand them. Thanks for the opportunity to express the views of Trust for America's Health today.
If the folks have just come in, just grab some food, you can find some open seats.
Glad you're able to join us today. I'm Jim Megan. I'm going to talk a little bit about diagnosis and detection technologies. Could we have the first slide, please? We commonly think of diagnosis as individually oriented toward a human, but in the context that we're using it for biodefense, we're using it on a broader detection diagnosis basis. So although it does help us understand what's going on in the body and allow us to develop antivirals to treat it or antimicrobials to treat it or vaccines to prevent it, in a larger sense, we need detection diagnosis technology to look at food, water, soil, all elements of the environment to determine when we have an attack, and of course to guide us in our response to that attack. So what are these technologies used for before the attack? Obviously, as you can see here, monitoring humans, animals, air, water, to get an early awareness of the attack. But in addition, diagnosis is used to look at the body's response, either a model animal or a human, a response to these agents, because many of these agents are esoteric. We don't have a lot of knowledge about how to treat them or to prevent them. In addition, it's important to establish a baseline of what's occurring in the environment. In North America, there is plague and there is anthrax naturally occurring. And so detecting and establishing what those baselines will be is very important. After the attack, diagnostic and the detection technology is important to determine if what the agent actually is down to the strain level, or determine whether it's been genetically modified. In addition, it helps guide us with therapeutics for vaccines, especially when they're limited. If we only have a certain amount in the stockpile, do we give it to a particular city or don't give it, depending upon whether we can detect the organism there. So it's important in determining the extent of the attack and establishing how it spreads. And in addition, helps us in cleanup. In a building like this, you wouldn't be able to vacuum up all of the spores, but you would be able to, in essence, fumigate it. And in that case, being able to detect whether the organisms are alive or dead is very important. And finally, for forensic purposes as well, to establish who the bioterrorist was and what strain they were using. This is a rough cartoon of what would go on. There'd be an attack, as you see here. There'd be some sort of an alarm or trigger, and that could either be a red light on a machine down in the metro, or it could be a cluster of cases in a hospital, or perhaps an outbreak on a farm. And that would lead to collection of samples. Oftentimes, the first responders, the people who are far forward, do a simple test, and then pass the samples on to a more sophisticated laboratory for identification and reporting. There are two basic approaches to diagnosis that are common now. If this is an organism on the right here with an internal nucleic acid and an external coat. One set of approaches is based on PCR technology. You've probably heard that, polymerase chain reaction. There are a number of varieties to this test. And it basically tests, in this case, the DNA inside the organism. You collect the organism, crack it open, and look for the DNA. It's very sensitive, very, very sensitive, very specific, but it's right now somewhat slower, has complex equipment, and it won't detect toxins. Toxins, as you remember, are excreted from many microorganisms, almost in a water or a poison form, and they don't have any nucleic acid. So that's a limitation for this type of technology. The second is based upon immunoassays, antibody-based assays, and a variation of that is the pregnancy kit that many of you have seen with a line that turns positive. These sorts of assays detect the outer coat of the antibody, not of the organism, not the nucleic acid inside. And their advantages are cheap, they're rapid, they're portable, but they're about 100 or 1,000-fold less sensitive than the nucleic acid assays. These are the problems that we're facing now in doing research to improve detection and diagnosis. 4 key problems. First is that in most cases, if you want a highly sensitive test, you have to trade off speed and portability. The developers of the assays are always balancing these things. In addition, many of the assays that are out there now could be fooled if there was a genetically altered organism. Although we hope that will never occur, I think we're in the defending against bioterrorist mode for many generations. And it would be foolish to think that they won't become more sophisticated and able to alter organisms. So that's a concern as well. In addition, many times the organism is present in very low concentrations, sucked in down in the metro, 1 or 2 spores in a large amount of fluid, or oftentimes it's present in a very complex matrix. Food, for example, chocolate ice cream is a particularly difficult thing to detect organisms in. So the research that's going on now is addressing these issues. And one of the major approaches right now to improve on the nucleic acid type of detection is to do rapid sequencing of organisms. So they're developing new methodologies where you can actually take an organism, crack it open, stretch out its DNA, and quickly determine the exact sequence of the organism. This helps us not only for specificity, but if there was a genetically altered organism, you'd be able to read it out right away. Some of the promising technology there is based upon a nano sensor, a nano hole, and they stretch out the DNA and put it through the hole, much like a string of pearls going through a ring. And as each one goes through, you read the base and read it off. Another major area addressing this is miniaturization. Much of the nucleic acid, much of the immunoassays are being adapted to nanotechnology, a word that you've heard, and being able to use very small surfaces where, in a relative sense, the sample is exposed to the surface and there's a large or a very fast ability to do reactions all across that surface. So you get a lot of speed out of that, you get the ability to move the sample through various channels. And so rapid sequencing and miniaturization moving toward nanotechnology are the 2 major thrust areas. And I wanted to just show you one example of that on the next slide. In nanotechnology, you're able to put very small amounts of material down on reaction surfaces. This is a slide, a 1x3 slide that you all know. And in current format, you can put 8,000 spots, both either DNA or protein on them. And this is an advantage in this particular case. If you were infected with anthrax here today, you probably wouldn't develop a fever for a few days. But prior to that symptom developing, your body would be fighting the organism, and the organism would be replicating in you. So if you could take some serum from you and look and get a fingerprint of what was going on in your body well before you showed a fever or a clinical sign, that would help us quite a bit. So looking at rapid host response is a major area of research. And my final slide just reviews some of the problems we have, enhancing the research effort to control, get very fast, accurate, easy assays in the future. One of the problems is that the funding is spread amongst a number of agencies, and the coordination is a little bit of a problem there. And some of the areas as well, such as detection of plant material or detection of animals, hasn't been well funded.
Hello, I'm delighted to see everyone here. I'm Una Ryan. I'm the CEO of Avant Immunotherapeutics, but I'm here to represent all of the vaccine companies in bio. The next, please. Now, in the infectious disease space, we're faced currently with new emerging diseases, with re-emerging diseases, and deliberately emerging diseases. And we're going to focus this morning on the deliberately emerging diseases, but I just want to remind you that we have re-emerging diseases because there are organisms that have become resistant to the drugs that we've had historically for them. And so I'm going to make a great case that for all of those categories, what we really need are vaccines, because vaccines have been responsible for wiping out many diseases and doing so in a permanent way. Now, in the deliberately emerging disease space, the government has come up with a list of priorities of threats, categories A, B, and C. And the category A threats are those which will tend to kill you most easily and which can be most easily weaponized. And the category A contains things like anthrax, botulism, plague, smallpox, tularemia, and the viral hemorrhagic diseases. And categories B and C are only slightly less scary. So what we really need are vaccines for all of them. And what we have, we actually so far have only managed to stockpile vaccines for 2 category A threats, anthrax and smallpox. And so I'm going to spend a little bit of time telling you why, with all the capabilities in biotechnology, there really aren't more vaccines that have been developed. And if there's one thing I leave you with, I want you to recognize that developing vaccines is a very long process, a very expensive process, and a very risky process. And that's really what stands in our way, and that's where we really need to share the risk with the government. So the good news is that we have new understanding of the basic principles of immunology, molecular immunology, which allows us to design very much better vaccines than we had in the past. And we have new technologies which allow us to make them more easily, deliver them more easily, and make them more effective more quickly and with lower doses. And so I think we're poised to be able to bring forward a very large number of new vaccines. The next, please. There are 2 sort of categories where I think biotechnology can help. We can take the more old-fashioned vaccines, the ones that have been stockpiled already, where the vaccine technology has been around, I think Rich said, for probably a century. My grandfather was in this business too. And we can make them more safe and more effective, and especially we can tailor them for a very rapid response, because in an emergency, you're not really looking for lifelong protection. You're looking to get through the crisis. So slightly different rules. But the place where it's so exciting is the emergence of the really novel technologies. We can change vaccine thinking completely. For example, most people think of a vaccine as a shot, and that's not really the ideal way to deliver mass vaccines for the whole nation. If you can start to think about vaccines that are needle-free, single dose, that protect very rapidly in days rather than in weeks or months, that are easy to distribute. For example, they are temperature stable and don't have to be refrigerated or frozen, and above all are easy to make, by which I mean probably cheap to make and cheap to distribute. All that you can get from biotech. But with that great stable of opportunities, there are a number of things that we are going to require in order to persuade people to move these things forward. There are lots of technologies and very, very promising opportunities that are kind of stuck in people's pipelines. They can take them through animal studies, or they can take them through the first benchtop experiments, but they can't afford or won't afford to take them all the way through to being products. So the first is guaranteed purchase. And that really means we have to have a guaranteed market. And if the government is the market, we have got to know how many doses are needed and what we will be paid for those doses. In addition, you've heard about the Valley of Death. Being from Boston, I used to call this a pothole, but it's grown, it's now a valley. And this is really what the very high-risk, very expensive area between the kind of funding you get for research from the NIH up until the time when you are ready to get a procurement award from the government. And this is really the most risky and the most expensive part of biotech development, and especially for vaccines. You need to do late-stage clinical trials, and these can often run in the thousands of patients. We've just done one that actually involved 86,000 patients, which I think is a world record, and I hope never happens again. But in addition, you have to have your manufacturing facility up to speed before you get paid. And you have to have predictable regulatory pathways. So this is all a way of saying, for us, and biotech likes risk, for us to be able to take on the risk, we need to be able to understand what the market will be at the end, understand the regulatory pathway, and get some help for those very risky middle years, and I mean years, 10 to 15 years and $1 billion to develop a vaccine. Next, please. So this is my last slide, and having told you all of the problems, I wanted to end on a happier note. These are the kinds of technologies that biotech companies are working on. Certainly the characteristic weakened, attenuated vaccines, but now single-dose oral vaccines that can be made easily, vaccines that can be made to give you T-cell responses, so you can give them after you're already sick, DNA vaccines, conjugate vaccines, both of which are easier to develop and give you increased vaccine responses, and the pure recombinant vaccines that are much safer and more predictable than the wholesale ones from the past. But we also have new technologies which will turbocharge the immune system. We call them adjuvants, and things that improve the vaccine response. And we shouldn't forget the new delivery systems which allow us to avoid needles and to get to more effective responses. So there's a wealth of vaccine technology out there. We simply need to share the burden of risk, and we'll get them to you. Thanks.
Good afternoon. I'm Dennis Ruby of Biotechnology. I'd like to discuss therapeutics with you. Now, in addition to vaccines, the development of therapeutics is an essential component of our defense against category agents. And there's a number of reasons for this. First of all, as I mentioned, there are not vaccines available for all these agents. Second of all, if they were, you could not vaccinate all the populace. And third of all, you need to be able to protect individuals who've been exposed, and there's not time to vaccinate them and develop a protective immune response. The approaches that are being used for drug development include the traditional high-throughput screening that has not been applied to these agents before, and a variety of newer state-of-the-art technologies as practiced by biotechnology companies. Now, Timmy from Elucis will be talking about antibacterials, so I'm going to confine my remarks to antivirals. Viral pathogens in category A list include several families of viruses that cause hemorrhagic fever, such as Ebola, that you're familiar with, and the orthopox viruses, such as smallpox. Of these, smallpox provides the greatest risk due to its ease of transmission and the severity of the disease it causes. If smallpox were introduced to the air handling system of this room or in the delicious lunch this morning, about 80 to 90% of you would become infected. First week or so, you think you have the common cold. at about 10 to 14 days. Next slide, please. The characteristic pock-like lesions would erupt. 30 to 40% of you would die. The remainder of you would bear the scars, both visible and invisible, for the rest of your lives. Next slide. In many respects, smallpox is the ideal bioweapon. It's easy to grow, it's easy to disseminate. A long incubation period facilitates its dissemination. Here in the U.S., we ceased vaccination in the 1970s, so most of you are susceptible. Although we're against stockpiling vaccine, it's not without its side effects. There's currently no approved antiviral drug. So as smallpox were put into our biospace, it's not a national catastrophe, it's a global catastrophe. Next slide. Our current defense options are relatively limited. They're quarantine, which is difficult to implement in today's mobile society, vaccination, treatment with protein therapeutics, or the development of antiviral drugs. Although vaccinations, go back. No, I'm missing one, that's okay. Go ahead. The slide I'm missing was simply to show you that vaccination using the current vaccine has serious side effects: myocarditis, encephalitis, and a variety of horrible skin infections. Although vaccination, we believe that antiviral will be important for a number of reasons. We can use it to prevent disease in non-vaccinated individuals, treat individuals previously exposed to smallpox, prevent disease in those populations you can't vaccinate, use in combination with vaccines to prevent smallpox and vaccine-related complications, and treat other orthopox disease outbreaks. We believe such a drug is on its way. ST-246 is an orally bioavailable drug. It's already shown excellent safety profile. It works in a number of animal models. An IND on this drug has been submitted and approved, and we're in the midst of human trials as we speak, and they're looking quite promising. Definitive animal challenge studies will begin shortly, with the intention of advancing to the NDA in 2008. Next slide. In addition to smallpox, we're working on therapeutics against the other Category A agents. As you can see from the slide, these are in various stages of advancement. Some of them should be advancing into clinical trials in the next year or so. To conclude, we believe the disease due to Category A agents is real. Whether they're delivered by a terrorist or, as others have mentioned, they're delivered by Mother Nature. It's going to happen, and we need to be prepared. Now, despite the availability of some vaccines, by and large, the U.S. populace remains at risk of these pathogens, and we believe that vaccines plus therapeutics provide the best defense option.
Good afternoon. I'm Tibi Casilico from Elucis Therapeutics. My former colleagues have given you a very good overview about the different technologies available to us through biotechnology, for vaccines, for detection methods, for new therapeutics. What I want to discuss with you today is to highlight some of the significant challenges that we in the biotech industry face in trying to meet the needs of the country's biodefense and bioprotection programs. And also, after that, emphasize some of our recommendations of the key areas where improvements can be made to help create a better partnership and help sustain the development of new biodefense measures long term. So the first slide. I'm going to use my company, Elucis, as a little bit of an example. We are a company that's focused on making targeted drugs to treat infectious disease. So we're a very good candidate to help participate in a partnership on biodefense development. We have technologies that can be applied for either commercial or biodefense applications. So we, strategically as a company, have to decide, are we going to invest our efforts into developing biodefense drugs, or are we going to move towards commercial markets? We've been involved since pre-9/11 at Elucis in the development of an anthrax therapeutic. So we've been around for a while. And in about 2002, if you were to look at our company pipeline, you would have seen 3 or 4 different biodefense targets that we expect to work on to develop countermeasures. Today, our pipeline only has one, and that's anthrax. And that's because of the fact that it's very difficult, after 5 years of working on this, it's still incredibly difficult to fund biodefense development, and the promise of very uncertain revenue is very hard for the financial markets and our board to get their arms around. So it's very hard for a company that's small, particularly in our situation, to move these programs forward without significant funding. The next slide, please. So we've been fortunate over the past 5 years. We've worked on our program. We've been supported with DoD funding and NIH funding. We've developed a very effective therapeutic. It's been tested in animals. It's been tested in people. It is ready to be manufactured for the Strategic National Stockpile. We've been able to develop this drug in only 5 years, and my colleagues and other companies have done, a handful of them have also done the same thing. We've moved very quickly in biotech to get these drugs developed. And so in the same time that we have developed an effective drug that is ready to be scaled up and put in the stockpile, there is still no effective procurement process to even purchase these drugs. So this is particularly important for those of you who were in the Hart Building in 2001. You know what it's like when there's a real scale and the drugs just aren't available, or if you had to take Cipro and that had negative effects. So the process is important. One of the most difficult things we've had, the next one, is finding out what the process is. This is a list of all the agencies that my company has interacted with in the last 5 years on multiple occasions. It's like alphabet soup, and it's an insane list, but we really had to talk to all these people to try to figure out who's on first, what's the process, who's in charge of it, who's got the money, who knows anything about BioShield. So this is a very difficult thing. It costs us a lot of time, but it's more important, it costs us a lot of money. Next slide, please. So I want to talk a little bit about sort of the realities of BioShield from our perspective and some of the roadblocks, and I'll follow that with some other examples of things, ways we think we can improve the process. The BioShield procurement process is still very undefined. We don't understand how it works in industry, and this is a very expensive process. We're trying to develop drugs that take 8 to 10 years to get ready for human use. This is a long-term commitment. It's a lot of money. What we need, it's very hard for us to commit to this without a certain commitment of purchase. We need to know that there's a buyer at the end of our development process. It's a little bit like building a battleship on speculation. That would never happen. And this is the same kind of dollars we're talking about. And we can't change every year and flip to flu and flip to SARS. If you commit to anthrax, you commit to a vaccine for Ebola, you've got to go the distance. In our case, the financial markets, which we depend on for funding, they don't value a potential contract. If you have a real contract, that's something they can get their arms around. So they're not supporting the funding of this program. So from our perspective, countermeasure development right now carries an enormous amount of risk, most of which is being carried by the biotechnology industry. And in order to have a partnership with government, we've got to share this risk. I'm going to talk a little bit about what people have alluded to, again, and Una did a nice job of it, the valley of death. What is the valley of death? Next slide. Okay, sure. I have a little more slide. And as Una rightly laid out for you, many of us get to this point. We have proof of concept. Many of us have gotten very close to being able to have a drug we can scale up. The problem is this process here is not funded by BioShield. It's not going to be funded by the biotech markets or the financial markets, and it costs $20 to $50 million to get your drug through this process to meet FDA's requirement to be purchased by BioShield. So for some of the things that we need a defined process. We need to know what the government wants us to make, and we need to know how to make it. We also need to know that there's a complete commitment to purchase these drugs, and we embark on this process and we take the risk. And as Una said, we are willing to take the risk. Adequate funding for the valley of death is critical. BART is a very good proposal, and we think it's a critical piece of getting through the valley of death issue. We need to have a clearly communicated procurement process with clear commitments for purchase. And in this way, we have a shared partnership with government where we share the risk and we also share the reward of getting new drugs into the stockpile. So my final slide is simply to say the biotech industry is willing to take the risk. We have the tools, we have the people, we have the skills. But in order to work effectively with government, we need to have sufficient funding, we need to have a defined process, and we need to have defined purchase procurements.
First of all, let's give our panelists a hand here. Thank you, of course. And now we have some time for any questions and answers that the audience may have. Feel free to ask any question you may have, and we have the esteemed panel up here to handle.
see, so you disaster. I mean, an aside there. I represent Concord Towns and Head District, and that's Brooklyn. And my constituents have 60% of them in healthcare now have terrible difficulties accessing the healthcare system in general, not to mention this kind of stuff. We held a port security meeting last week about some of these issues in New York. I mean, obviously, we're the ones that get hit by anything, put it where it is. I understand you're market concerned about getting your own products to market, but I can see you're in an environment where we can't even get a health information technology book behind us, you know? I mean, and I would really like to see some of your products in a demonstration project, which also connect the communities in an information technology environment, to see what happens, if they work, what's going on. But I mean, we got some real regular concerns in terms of health. I understand your needs as companies to get your products through to the end. But I mean, I can ask this almost like I'm a Republican. I'm going to say it anyway. I mean, those are your commercial concerns. Why should we, as a government, and why should we interact with that when we have other pressing healthcare concerns? Anyone?
Well, I think, for example, vaccines have shown time and time again that they're probably the most cost-effective type of medical treatment that we have today. And I think the dollars would be very well spent. I like your idea of demonstration projects and information.
We'll talk about that after. I mean, I'm serious about it, too.
No, I am too. But I think actually this could be a model for much more effective healthcare. I think vaccines are an incredibly effective way of protecting.
I think there's a tremendous amount of spillover. I mean, what's going on in the biodefense industry is really application of state-of-the-art technologies in diagnostics, vaccines, and therapeutics. And a lot of those drugs and vaccines that come out are going to be applicable to traditional diseases, and a lot of things we learn about how to distribute these will also be applicable. I mean, imagine if we had a drug available for HIV before the epidemic broke out, what sort of savings we would have as a nation. And that's just, we don't know when the next pox virus or Ebola virus is going to break out. But preparedness is going to save an awful lot of money and a lot of lives down the road.
Many of the dollars that have gone in have really shored up the public health community, which was in pretty tough shape. Your own city was the entry point for West Nile. And in that particular scenario, there were only 1.5 people who were doing mosquito control in New York City at that time. In Suffolk County, there were 12 people. So a lot of the money has spilled over already. If we have a rapid way to detect in mosquitoes or a rapid way to detect in anything, it's going to help the general population. A lot of the money went to public health groups who bought ELISA readers, who bought PCR equipment. When we first started this, I think it was only 5 state labs could do PCRs back about 5 years ago. So a lot of that is going to have tremendous spillover and raise all boats.
So PCR is again?
Polymerase chain reaction, one more modern methodology. So you're going to see a raise in all boats.
Sure.
Two of the major items on the wish list seem to be clarity and commitment from our part and funding. But what I didn't hear as a major concern was the regulatory process itself, not the clarity, but whether it's too stringent or what satisfaction or dissatisfaction there might be from that. And because I think at least some of our bosses are more open to the idea of deregulation than to the idea of cost sharing or risk sharing, and that would be easier to set.
I would say a considerable part of what we talk to in Valley of Death, regulatory issues are there for sure, but of course we want to make safe drugs. There are ways we could facilitate that process. However, the major costs are really making the drug. As Una said, we have to commit to the manufacturing, and we have to commit to practice lots of the manufacturing beforehand. So in the case of my company, we have to pay out $25 million just to be able to prove to the FDA that we can reproducibly make the drug. And I think that's probably, it's just a real cost. It's making it. The regulatory issues, I think, there can be some improvements there. but we still have to be making a safe book.
Anything specific?
Well, streamline regulatory processes, ones that are applicable to some of these new concepts we have. I mean, there's a tremendously sort of old way of thinking. You know, people will say to me, so how many injections and what adjuvant? We don't have any injections or any adjuvant. It's one sip and go. And yet the vectors we use are actually human pathogens, and so they don't work terribly well in mice and rats. And so we need much more scientific thinking in the regulatory process.
One final comment that the government has been at work, and the FDA has come up with what's called the animal rule. And that's the reason that today we have been able to make a drug in 5 years. Without that rule, we'd have to go another 5 to 6 years in development.
And that's my problem. I'd rather go straight to humans where my vaccines don't work.
Any other questions? Sure.
I apologize, I don't know as much about this as I should, but is there very much duplication of effort among the companies trying to develop these biodefense therapies?
I'll answer that. There are a lot of companies that start out and take advantage of NIH funding, and there's a lot of early stage research funding that is spent on a lot of companies, but very few companies to date have gone the distance, say that our company and Pharmathene and some of the others have gone go, to hang in there with this sort of uncertain process. So in the final analysis now, I don't think there's a lot of duplication. People are focusing on anthrax simply because the list has not really been clarified and the commitment to other pathogens is not yet made.
I think there should be more. Actually, I think we should have had more redundancy in the stockpiling and manufacturing. You know, if you have one manufacturer and that plant goes down, that's it. We saw that with flu. So I think actually it would have been better to have had 2 or 3 manufacturers for each thing.
When you spoke of the pothole, or the canyon that you were talking about, say you're in the middle of the process and you have a great idea, you have a great vaccine, and you're running short on money. Have you ever had to go to the government to get more funding for?
Bring on.
Bring it, we go.
You can try, but I really want to distinguish from the first question as well. We're not here asking for money to keep our companies afloat. We're here saying we need money to do these problems. I mean, I'll still make vaccines for travelers and all the other uses. So you can go and try if there's an RFP that would suit you, but
And the issue we're
there's
trying to
no place
bring
to
up
go.
is that BioShield put aside $5.6 billion. It's there that the companies and the industry and government felt we really need to protect ourselves with the Biodefense program. The problem is we don't have any ability to deliver those drugs because of the lack of the funding required for the value of death. And the real issue is here, we won't develop the drugs if we don't have the money to pay for them. We just will go develop other things.
I would interject right now to deal with the valley of death. There have been a couple legislative proposals that we're familiar with, which we believe are good bills, and those are on the Senate side. Senator Burr has a bill which deals with the valley of death and the creation of the Biodefense what's called BARDA. And on the House side, a similar bill has been introduced by Congressman Rogers, Mike Rogers from Michigan, and Congresswoman Anna Hession from California, which would do much the same. So there are some ideas out there to deal with this valley of death.
And this is part of the bill is really to redeploy some of the existing allocated funds. So it's not talking about new funds, it's just a better use of the existing funds to make this happen.
Yeah.
There's a very successful program, the Small Business Innovative Research Program, SBIR. So think of the development of any of these things as going from the laboratory bench all the way to the bottle. And if you have a good idea, you get funded from NIH and basic research at a university, they eventually spin off and often use these SBIR grants. But then many things die right after there. That's the start of the valley of death until at the other side of the valley, there's the procurement dollar. And it's that gap that we're talking about.
Any other questions? Well, again, thank you very much.
The real